In yesterday's class, we talked about cell cycle. We talked about how cells replicate or divide for growth and repair and undergo the process of mitosis, where one cell divides into two. It is a controlled process and it happens with all the cells during development and early growth, but when the organism becomes mature, many of the cells stop undergoing this process. Some do, and how often depends on the need, such as bones and the liver and the skin. There's also the act of asexual reproduction in plants and bacteria and other organisms. There's the hydra, which is it is a multicellular organism which also undergoes asexual reproduction. And so only one cell or one parent organism gives rise to a genetically identical offspring. Then there's sexual reproduction, which involves two parents and meiosis, which will come in later. We talked about chromosomes, which are present in the nucleus of the cell, and they are basically the DNA molecules, which are the genetic material. And chromosomes are the coiled and condensed structure that DNA forms after wrapping around histone proteins and further supercoiling. Chromosomes are only visible during cell division. So when a cell is actively dividing, these can be seen during microscopy. Otherwise, they cannot be made out as specific or clear structures in the nucleus. Every species has a characteristic number of chromosomes, and because they have the genetic material coming in from each parent, the chromosomes form pairs. So in humans, there are a total of 46, which gives rise to 23 pairs. 23 coming in from the mother and the other 23 from the father. Of these, 22 are autosomes, and there's one pair of sex chromosomes. We talked about the karyotype, which is a picture of all the chromosomes. Then coming to the actual cell cycle, it is the process, as you mentioned, of cell division replication. It starts off at interphase, which is divided into G1, S, and G2 phases. All non-dividing cells are in interphase, and specific, the G1 or the gap1 stage of interphase, where they grow, carry out metabolic activities. When they receive signals for division, they enter the S phase, where DNA replication occurs. And so each chromosome doubles itself, so to speak, and forms two chromatids joined together. So this is a chromosome in its replicated state, and then goes on to the G2, where other organelles and required substances are replicated and gathered, and a lot of synthesis taking place. Then briefly, mitosis is where nucleus or genetic material is separated into two. Cytokinesis is the cell division, physical cell division, where one cell physically separates into two daughter cells. It is a well-controlled process. There are checkpoints, and they're regulated by small proteins called cyclins, which bind to enzymes called cyclin-dependent kinases or CDKs. And this cyclin-dependent kinase cyclin-CDK complex, it phosphorylates other proteins, changing their shape and therefore acting as signals or checkpoints for the next stage or next step of the cell cycle. Examples are phosphorylation of chromatin, causing them to condense further, and phosphorylation of proteins in the nuclear membrane, causing breakdown of the nuclear membrane during cell division. Now today All right, so that's an overview. Today we will be talking about mitosis, and if time permits, we will carry on to talk about stem cells. So this is chapter 3B2. And this is the stage of the cell cycle after interphase. Right, it is also sometimes called the M phase, M meaning mitosis. And we'll see, obviously, there's a lot of activity going on in the cell, and a lot of ATP is produced and utilized. And so there are 4 stages of mitosis. Those are we're starting with what is called the prophase. That's one word. So prophase, metaphase, anaphase, and telophase. So let's start off with prophase, obviously. So the genetic material has already replicated. During the S phase of interphase. And what we will see over here is right, the chromosomes, right, they coil up, and they have the 2 chromatids as we just saw in our review and yesterday's class. And there's right the point of attachment. If I just go back, the point of attachment, this green sphere that I've drawn for the 2 chromatids, is called the centromere. I'll just add that over here. Right, this term will be used in today's explanations, so I'm just adding that. So what happens over here is the nucleolus. The nucleolus is right an extra dense part within the nucleus. Nucleolus, it breaks down. And the centromeres. Right, so these are also organelles, centromeres. They move to opposite ends of the cell. right? So that's what's happened over here. In the metaphase, the nuclear membrane, so right, that's the beginning, that's the prophase, then you come to the metaphase, and the nuclear membrane breaks up. Right, the centrioles have moved to the opposite poles of the cell. Centrioles are at opposite poles of cell. And right from each of the centriole, right, there are microtubules that are formed, and they move all the way to the chromatids, forming a spindle. So they form a spindle of microtubules that attach to this centromere, to the centromere. I think I've confused the terminology over here. Yeah, so these are centrioles. So at the centromeres, so that is at the point of attachment for the two chromatids, the replicated state of the chromosome. And right, the chromosomes, all 46 of them, are arranged along the metaphase equator. Alright, so the cell looks something like this during metaphase. Alright, you've got your chromosomes. In fact, draw them like this. So these are chromosomes with their chromatids, and you've got their centromeres over here. The green isn't really visible over the blue. And you've got your centrioles. And they form spindles. Right, so these are microtubules that go all the way and connect to the centromeres of each of the chromosome. Okay. Point over here is, right, this is basically explaining how it happens in mammalian cell. Plant cells undergoing mitosis, so there's no spindle forming. Because they do not have centrioles. So since plants don't have the organelle that produces the spindle, they don't have a spindle, obviously. That's on the side note. Okay. Then we come to anaphase. And during anaphase, the spindles pull on the centromeres, and separate the daughter. I haven't written down this term, even though I've used it during explanation. The daughter chromatids. So again, if we just draw that, right, you've got chromosome that's replicated, right, and that's the centromere. And we've got spindles coming in from both ends, and they're pulling. And so what you get is you have, right, the chromatids separate. And so one copy of each chromosome, so one copy of each chromosome moves to either pole of the cell, right? Either pole or either end of the cell. Right, and this is a very quick process. And so, right, you've got a full set of chromosomes at each end of the cell. Now, at this point, they explain that the spindles are formed with overlapping microtubules, and spindles are formed by overlapping microtubules, and they contain contractile fibers. Contractile fibers. Right? These contractile fibers are similar to those in animal muscles. Right? And so these fibers, the contraction of these fibers are what cause the movement of the chromatids. And this is a process that requires ATP. Then we come finally to the last part of mitosis. It's called the telophase. And over here, right? The spindle breaks down, the nuclear envelope reforms. And the chromosomes begin to decondense. And the centriole is reformed. So why are they saying the centriole is reformed? Because in the cell, right, in a normal cell, that is not dividing, you have the centriole structure at some place in the cell, something like this, right? When the cell divides, right, these two move apart as, right? So I drew the whole thing over here. If I make it accurately, then the way it looks is perhaps just a single cylinder type structure, right? So when this happens, right, the centrioles had moved apart, right? One each formed the spindle, pulled the chromatids apart, and when that's all done in the telophase, they come back together. Okay, what else do we have? That's all, right? So at this point, mitosis is complete, right? But the cells have not yet divided. So what you're going to have next is called cyto cytokinesis. This is the final stage of the cell cycle. And it's basically, right, the division of the cytoplasm. So what happens in animal cells is, right, we'll talk about animal and plant cells a bit separately. It's slightly different. So in animal cells, there is a ring of contractile fibers, right? It tightens around the center of the cell like a belt. Right, again, these fibers are similar to those found in muscles, and right, they continue to contract until, right, a cell has been split into two, or two cells have been separated. Okay. In plant cells, right, uh, because they have cell walls, right? So what happens is uh, the cell wall starts building up from the inside to the outside of cell. Right, and so you've got one cell and there's a cell wall building over here, and that separates the two daughter cells. Right, because they're pressed together, perhaps you can't make them out. So that's how one separates into two. Right, and also in the case of plant cells, right, so the materials for the cell wall are obviously transferred via vesicles because it's going from the inside to the out. So materials for cell wall transferred via vesicles. And so what happens is that at certain point there are gaps, right, in cell wall, right, where there weren't any vesicles, and these form a structure known as plasmodesmata. This is the plural. The singular is plasmodesma, and these are regions of the cell wall, right, that I can only make it once over here. So there's a gap in the cell wall, and this allows cytoplasmic connections between neighboring cells. And so it's not just here, right, between the new cells, it's between all neighboring cells. Obviously not as large, right? And so plant cells are connected to each other through their cytoplasm as well, through these small pores. And we come to know that they're actually formed during cell division when new cells are being made. In either case, right, you have one cell dividing into two, into two genetically identical daughter cells, right? And once this is complete, they go back into interphase. And by that, right, we're talking about G1. So whether they continue on dividing or whether they just grow in size, right, and start carrying out their metabolic activities, that depends on whether there's a need and whatever signal is being given. Any questions?
No, sir.
Right, so you can find diagrammatic representations of all of these stages in the book. Uh, they talk about the importance of mitosis after this, which is we already covered. There's the asexual reproduction part, which produces genetically identical individuals, or clones is the term they use. And right, so advantages, I just note that down here pretty quick. Of mitosis, you have asexual reproduction, you get genetically identical clones, right? This is a rapid process. As you mentioned, does not need two parents. This also becomes a problem because there is no variation. Right, so the entire population can be affected and possibly wiped out by any changes in environment. Or, for example, a disease, because they're all the same genetically, and so they're all going to come under effect. And there are species such as like fungi or plants that can, reproduce both sexually and asexually. And lastly, they talk about something called the mitotic index. So this, as they mention, is a measure of how actively cells of a tissue are divided. And how they figure that out is that they take a sample and they stain it and see the number of cells visible total. So you take a sample, staining, and observe, right? You observe the total number of visible cells and the number of cells in mitosis. I'll show you a picture of this where the cells, the nuclei can clearly be seen, right? The chromosomes, well, not the nuclei, the chromosomes and the nuclei can be clearly seen, right? And so they're undergoing mitosis, and so they measure the mitotic index as the number of cells in mitosis divided by total number of cells. Now there's a use for this, right? So identify actively dividing tissue. That's the whole point. And it can also be used to identify cancerous tissue. So if a tissue has an abnormally high mitotic index, or it is right, it has a lot of you know actively dividing cells, and especially when it shouldn't, that means it's probably cancerous. Right. So not only to identify cancerous tissue, but also cancer treatment. So when you're treating for cancer, you're going to check that tissue again and see whether the mitotic index comes down. Right. So the mitotic index in this case should lower because of the treatment. So that means the cancer cells are being killed off. Right. These guys have unusually high mitotic index, activity, mitotic index for that tissue. Right. For example, someone has a liver cancer. Liver cells don't just go around dividing, having very high activity in that domain. They are very, very active cells, but not in the case of mitosis. They're not always dividing. And so if that is identified, then it might be a case of liver cancer, for example. I'll just take you to the book, show you a picture of this. All right, so here you can see this is for plant cells, this one over here as well. All right, plant cells are easily studyable, and why are they plant cells? Because you can see the regular shape. And so, right, they are this shows the different stages of mitosis. You got right some chromosomes that are arranging, some being pulled apart, right? And this image is used for the mitotic index. You calculate the total number of cells visible. You can see mitosis happening here, for example, and here, and clearly over here. So that's anaphase happening over there, right here as well. So a lot of activity, and so you count the total number and those that you can identify as clearly being in the process of mitosis. You feed them into this formula, and you get the mitotic index. All right, and that brings us to the end of the chapter. Now, obviously, depending on the kind of tissue that you are talking about, right, would depend on, right. So there are reference values for mitotic indices for the type of tissue that you are studying. For example, the skin is generally actively dividing, so it will obviously have a higher mitotic index than, for example, your liver. Right? Any questions?
No, sir.
Right. So, seeing that we're just about
Why
out
are
of time, how about we leave stem cells for next time? Does that work? Right. It is still chapter 3. We're just going to skip this part where they talk about sexual reproduction and genes. As I mentioned, that part is in the A2 syllabus for Cambridge, so we'll circle back to that later. And
okay, sir.
right, we're going to move on to 3C4, which is stem cells, and 3C5, which is the use of stem cells. So how about we leave that for next time? Would that work?
Yes, sir. No problem.
Okay. So I'll see you next class then, inshallah.
Yes, sir.